of hosting today, and we are incredibly thrilled and delighted that we were able to induce Dr. Gigi Lozano to be our speaker today. Dr. Lozano graduated from Pan American University in Texas, did some work at Oak Ridge Laboratories, got her PhD at Rutgers, and then went to Princeton, where she was a postdoc with Arnie Levine. Immediately following that, she was recruited to MD Anderson, and in her long tenure there, she has done path-breaking work on p53. She's recognized for enormous contributions that include the recognition that p53 works as a transcriptional activator, many findings regarding the ways that Mdm2 and Mdm4 regulate p53, extensive use of transgenic models to understand these mechanisms better, definition of specific p53 loss and gain of function mutations and their effects on p53's biology, as well as studies of the p53 transcriptional program. She's a member of the National Academy of Sciences and the National Academy of Medicine. And at MD Anderson, she is now chair of the Department of Genetics and Hubert L. Olive Stringer Distinguished Chair in Oncology in the Division of Basic Science Research at MD Anderson. So I don't want to talk for a long time because I'm really eager to hear what you have to say. So thank you very much for joining us. And I will just say to the audience, you can use the chat function to enter your questions, or we can, I believe we can unmute people at the end if we need to. So with that, Gigi, welcome.
Thank you. Thank you very much, Barbara, for that introduction. I hopefully now am sharing my screen. Can you see it?
We can.
Okay, super. All right, so anyway, thank you, Barbara. It's fun to always visit a place, and now during COVID, we're doing these virtual seminars. But hopefully at the end, I'll be able to address some of the questions that people might have. What I thought I would do today is describe some of the models that I think have defined some of the basic, a real basic understanding of the p53 tumor suppressor pathway. So I'll get started with my disclosures. I am on the scientific advisory board for PMV Pharma. So the p53 pathway, this is my myopic view of the pathway. And it really highlights, I think, some of the critical features of the pathway. First and foremost, p53 is present at very low levels in normal cells, but any kind of abnormality that the cell senses, hypoxia, DNA damage, inappropriate activation of an oncogene, will stabilize that p53 protein, and then p53 in turn functions as a transcription factor to activate hundreds of genes. And I'll show you some of those experiments in a little bit. But some of the genes that p53 is known to activate is p21, for example, which is an inhibitor of the cell cycle. It also induces a senescence program. p53 can activate a slew of genes that can initiate apoptosis, and it also activates genes that are involved in changing the metabolic functions of a cell. Now, when p53 is activated, if the cell is allowed to survive and proceed, p53 has to activate this protein called Mdm2, which is an E3 ubiquitin ligase that targets p53 for degradation and basically removes the p53 levels back down to normal. So if you just think about what p53 can do, it can do so much. It can arrest a cell, it can kill the cell, and it can also induce its own inhibitor to allow the cell to survive. And even though there's so much work in the p53 field, we still don't understand all of the cues that determine which of these pathways p53 can activate. But because it has all these functions, it is a critical tumor suppressor, and it is the most often mutated gene in human cancers. So what I show here is what the field calls a Manhattan plot, and it was developed by Magali Olivier. So across this axis are 125 genes that are commonly mutated in cancers, and then across this axis here are 36 different types of cancers. And there's some features that stand out, but this is the one I want to highlight here. Across the board, these are mutations in the p53 tumor suppressor. So almost all cancers mutate to p53. But p53 pathway is inactivated by multiple mechanisms. And I show here some of the different cancers and how they inactivate p53. So high-grade serous ovarian carcinomas, mutations in p53 are the most common. However, in liposarcomas, it is upregulation of Mdm2, the p53 ubiquitin ligase, and about 100% of these liposarcomas inactivate the pathway by overexpressing Mdm2. And then here I show glioblastoma because we've become interested in glioblastomas recently. The p53 gene is altered, deleted in approximately a third of glioblastomas, and the Mdm2 and Mdm4 genes are upregulated in about a quarter. And this is a mutually exclusive relationship. So if p53 is mutant, Mdm2 doesn't have to be upregulated. And the other thing I want to point out in glioblastomas is that we have about half of these tumors that have neither mutations in p53 or upregulation of the Mdm2 and Mdm4 inhibitors of p53. And so since I really believe that the p53 pathway has to be undermined in the development of all cancers, I think that there's a big hole here that we have to understand in more detail. So today's talk is really going to concentrate on just a few proteins, and I'm not going to have time to show you a whole lot of data, but I thought I would use this slide to highlight some of the important functions that of these proteins that I will discuss with you today. So Mdm2 is an inhibitor of p53. It's an E3 ubiquitin ligase that targets p53 for degradation. Mdm4 also inhibits p53. It doesn't have any E3 ligase function, but it actually facilitates and makes Mdm2 a better E3 ubiquitin ligase, although it also has independent functions of Mdm2 and can actually bind and inhibit the p53 transactivation domain. This relationship, this Mdm2 and Mdm4 also can form a heterodimer, and that heterodimer is critical in embryo development to inhibit p53. And then, as I indicated and we'll discuss in some detail, p53 can activate the Mdm2 promoter, so it can upregulate Mdm2 and inhibit its own levels. Another important concept that I'll mention very briefly, maybe at the end, is that Mdm2 can also inhibit a mutant p53 protein. And that's because these mutant proteins have mutations in the DNA binding domain, but retain a transcriptional activation domain. But the important point that I want to make here is even though this mutant can be targeted by Mdm2 and Mdm4, it is mutant and so can no longer feed back and upregulate Mdm2. So with time, in our in vivo models, we find that these p53 mutants can become stabilized because there's insufficient Mdm2 to down-modulate the protein levels. And in a few minutes, you'll see how important that down modulation is. So the outline of my talk today is I'm going to talk about some of the models that showed us how exquisitely sensitive p53 is to inhibition by Mdm2. I'm going to tell you what the molecular response is to p53 activation in vivo. And last but not least, I'm going to describe some of the new cancer models that we're working with that express mutant p53 proteins. So let's first talk about the MDM proteins and their inhibition of p53. So a long time ago now, we attempted to make an Mdm2 null mouse, and it's just not possible. And the reason it's not possible is because an Mdm2 null embryo just prior to implantation is apoptotic. This embryo is stained with a tunnel assay, and every cell in this embryo is dead. And at the time, we knew that Mdm2 interacted with p53, but we really didn't know how important the interaction was. And what we did was test the importance of p53 in this little embryo by crossing to a p53 null mouse. And we completely rescue this phenotype. These mice are born and they're perfectly normal, except now because they lack p53, they have a tumor phenotype. So what this experiment indicates is that what MDM2 is doing in these embryos is upregulating p53, which is preventing the normal development of these embryos. MDM4 is a related MDM2 protein that Art Johansson discovered. And since MDM2 has such a unique relationship with p53, we decided that we would make the MDM4 knockout, but weren't sure what to expect. But an MDM4 knockout is also embryo lethal a few days after the MDM2 null, but again, that phenotype is rescued by deletion of p53. And we've also made mice that have no MDM2, no MDM4, no p53, and these mice are fine. They're viable. They have p53 tumor phenotypes because they lack p53. So at least physiologically in the mouse, the two most important functions of these MDM proteins is to keep p53 levels low during homeostasis. Okay, so we have these MDM2 heterozygous and these MDM4 heterozygous mice. They have only one allele of each of these two genes, and these mice are perfectly normal and running around. Okay, but as I indicated in my introductory slide, p53 is a DNA damage response protein, and so we wondered if there were any phenotypes in these mice if we irradiated them, if we damaged their DNA. And sure enough, we saw a beautiful phenotype. So the MDM2 and the MDM4 heterozygous mice are sensitive to low-dose ionizing radiation. So in this experiment, what we've done is irradiated mice with 6-Gray. And the black line above here is a normal mouse that for 50 days just ignores 6-Gray radiation. But the heterozygous mice, MDM4 in red and MDM2 in blue, are dead by 3 weeks of age. Importantly, if we now remove p53 from this system, we completely rescue these phenotypes. So this is the rescue of the MDM2 heterozygous mouse, and here's the rescue of the MDM4 heterozygous mice. So even though the MDM2 and MDM4 heterozygous mice have sufficient levels of these MDM proteins to maintain homeostasis, with damage, there's just not enough of these proteins to return p53 back to normal levels. So the next experiment that I want to tell you about is the importance of this feedback loop. So as I indicated, MDM2 is regulated by p53. There are 2 p53 binding sites in the P2 promoter that are p53 dependent. So p53 binds these sequences and activates MDM2. And so what we decided to do is to ask how important was this feedback loop in regulating p53 levels, okay? So we made point mutations, and I show here the different point mutations because we didn't want to disrupt the architecture of the promoter. We just wanted to disrupt p53 binding to the MDM2 promoter. So these experiments in the bottom are chip data that basically show that p53 cannot bind this mutant promoter, which we call P2P2. And this is a different promoter that shows our assays are working, and p53 can bind the Puma promoter. Okay, so we made these point mutations, and we were surprised that our mouse was perfectly normal. We really thought that this feedback loop was going to be critical for regulation of p53, but the mice are fine. But again, as in the previous case, they're exquisitely sensitive to radiation. So this is the same experiment that I showed you before. We've irradiated the mice with 6-Gray. Normal mice, MDM2 heterozygotes, don't care about this dose, but you can see that the P2P2 homozygous mice that no longer have this feedback loop are dead for the most part. So what is the phenotype? These animals are actually dying because of a complete ablation of the bone marrow. So here's a heterozygous irradiated mice, and you can see that at 12 days you have some disruption of bone marrow function, but it's still viable. whereas in the mice that lack the feedback loop, there's a complete ablation of hematopoiesis. And this is, again, a p53 dependent process. So if we take out just one allele of p53, we completely rescue this phenotype. So we can rescue this phenotype with complete deletion of p53 or heterozygosity of p53. So what these experiments are beginning to tell us is that there's this important relationship between Mdm2 and p53, and then there's an important balance that has to be maintained for survival after DNA damage. The last experiment that I'll show you here is our attempts to try to understand which pathway downstream of p53 is important for this phenotype. So I've already told you that heterozygosity of p53 rescues the phenotype. We also generated mice with deletion of p21, which is the cell cycle inhibitor, and that had no effect on the phenotype. Those mice are also very sensitive to radiation. And then we also deleted PUMA, which is one of the apoptotic targets of p53. And here you can see that there was a complete rescue of this hematopoietic defect. So in this scenario, it appears that it is the apoptotic function of p53 that is killing these hematopoietic stem cells. So those are just a couple of the numerous experiments we've done to evaluate the relationship between Mdm2, 4, and p53, and it's just an exquisite relationship. You need sufficient Mdm2 for survival, and if you have too much Mdm2 or Mdm4, deletion of p53, deletion of downstream effectors of p53 can rescue those lethal phenotypes. Now, because Mdm2 is so lethal early during embryogenesis, one of the experiments we wanted to do is really ask about an adult mouse and how important is Mdm2 in different tissues at different times. And we've generated this conditional allele of Mdm2, and this is using the LOX-P system. So these two LOX-P sites encompass two of the exons that code for the major binding region to p53. So this conditional allele then allows us to delete Mdm2 in any tissue that we want to, and we've generated a number of tissues and experiments that way. But what I want to show you is what happens when you globally remove Mdm2 in the whole mouse. So as the title says, Mdm2 loss in the adult results in a lethal phenotype. So what we've done here is we've used a Cre transgene that is tamoxifen inducible. So this is a mouse that has one of the conditional alleles, and it has the other allele is missing. So a single recombination event is going to create an Mdm2 null mouse or null cell. And all we do is inject tamoxifen, and then we look at what happens to these phenotypes. And I think you can see from this graph here on the right that within 5 days of treating the mice with tamoxifen, they're all dead. So losing Mdm2, and it's a p53 dependent process, causes this enormous physiological response. These are some of the pathologies we see in these mice. The hippocampus has a less number of cells. The retina is comprised of multiple beautiful layers, and you can see that in all of these cases, it's a decreased cell numbers. There's some differences in the liver, and it's actually, if you measure liver function is compromised when you have deletions of Mdm2. Kidney has all these protein casts and dilated tubules, and then in the spleen we have complete absence of white pulp. And as I indicated, these phenotypes are all p53 dependent. So I think the important aspect of this slide is that, in some of the previous slides, is it highlights some of the pathologies that we might see when we use Mdm2 inhibitors in the clinic to inhibit the tumors that have high levels of MDM2. And the hematopoietic defect is actually observed in human patients treated with MDM2 inhibitors. But now I want to use this model system to understand what P53 is doing in these different tissues. And one reason for doing this is we would like to be able to reactivate P53 somehow, and it's kind of hard. A lot of people are trying to reactivate P53. But what we were hoping is that we might be able to identify downstream pathways to P53 that would be better targets for reactivation in tumors. So let me show you what we did. So again, we used our MDM2 conditional mouse, and we deleted MDM2 in the adult mouse, but we did this acutely, and we actually chose a 24-hour time point to ask what P53 targets are regulated in different tissues that lead to these pathologies in the adult mouse. Okay, so this is now all the different tissues that we initially looked at. And what I'm showing you here is the percent recombination. So once we treat with tamoxifen, we induce recombination of the locus. And you can see that the pancreas, the heart, the intestine had the highest level of recombination. Again, I will point out that we only have one MDM2 allele. The other allele is a null allele. So a single recombination event will activate P53. And then on this axis, we chose to look at P53 activation by measuring the expression of P21, which encodes a cell cycle inhibitor. So you can see in this experiment that the kidney, the pancreas, and the intestine were the tissues that expressed the highest levels of P21. And we were thinking the highest levels of P53. Okay, so we've looked at these mice. So in 24 hours, we see no, so let me back up just for 1 second. I hope I can do that. So the tissues that we decided to look at were the kidney, the pancreas, the intestine, and the heart, and the ovary. And the ovary, just because P53 is mutant in ovarian carcinomas at 95% frequency. And so we wondered if we might be able to begin to understand that mutation frequency. So for the heart, for the ovary, we saw absolutely no phenotypes after 24 hours post-deletion of MDM2. In the intestine, we saw a fascinating phenotype, which is this crypt dropout phenotype. In yellow here, I've outlined the crypts of the intestine, and one of the phenotypes is the complete absence of a crypt. Sydney in the lab has quantified the number of crypts in these different animals, and you can see the mice that have no MDM2 have about about half of the number of crypts as a normal control mouse. The kidney also had some phenotypes at 24 hours, and it had twice the number of protein casts, so you can see here. So this is an early phenotype in the kidney. Again, this is the normal kidney control experiment. And then the pancreas had, to me, a fascinating phenotype, which we'll delve into a little bit more deeply. But what we saw in the pancreas is this acinar to ductal metaplasia. So here's a normal pancreas in the top, and here's what the pancreas looks like in the animals that have no MDM2. Here we've stained with keratin-19, which is a marker for a ductal cell. And here we've measured the metaplastic area, and we also see an immune infiltration in these mice. So within 24 hours, we saw this huge plasticity in the pancreas from this acinar to ductal metaplasia. Okay, so we've taken these 5 tissues and we've looked for expression of P53 targets. So on the left here, I show you all of the genes that were regulated in these different tissues in our RNA-seq data. The dark region is the region that is upregulated, and the lighter color shows the regions that were downregulated in each of these 5 tissues. On the right here, I show the percent of these dysregulated genes that are actually p53 targets. They have a p53 binding site. And we used data from the literature to identify these genes with p53 binding sites. So in the intestine, for example, I think that number is 69% of the genes had p53 binding sites. So the most of the genes dysregulated in the system by deletion of Mdm2 are p53 targets. The heart, on the other hand, had a huge physiological response, 604 genes that were dysregulated, but only 16% are p53 targets. So what we're capturing here at 24 hours is not just activation of p53 and p53 targets, but the downstream response to that p53 activation. So this is now a compilation of all those 5 different tissues to examine the overlap in p53 target genes. And as you can see from this figure on the left, there were only 7 genes that were commonly regulated by Mdm2 loss that are p53 targets. So for example, here in the pancreas, we have 206 genes. 135 of these p53 targets are specific to the pancreas, and 7 were shared with the other 4 tissues. So these 7 genes are Mdm2, cyclin G1, Mdm2, as I mentioned at the very beginning, is regulated by p53, and we expected, well, we didn't expect Mdm2, but it's not a surprising result because the promoter is intact in Mdm2. 3 of these genes, cyclin G1, GSTE1, PSARC1, are self-cycle regulators. 2 of these genes are transcription factors, and this gene EDA2R are cell death genes. So these are the 6 p53 targets that have a common, that represent the common signature of upregulated genes in these 3 tissues. We wanted to validate the signature to make sure that they were truly p53 targets physiologically. And so what we did is we did our DNA damage ionizing radiation experiment. We treated the whole animal with ionizing radiation. And here's the data for 2 of the genes, and we've done it for all 7, cyclin G1, EDA2R. Here's the wild type levels of expression of these genes. If we irradiate, you can see that these genes are upregulated in both cases. And if we irradiate a p53 null, you see no upregulation. So these are p53 target genes that are being upregulated following ionizing radiation. So these experiments highlight this incredible repertoire of transcriptional targets that p53 physiologically regulates in vivo. And I think it also suggests that maybe these specific targets can be used to understand in vivo if you have, if you can reactivate p53 or convert mutant to wild type. These might be great markers to look at for activation of p53. Okay, I want to now just briefly discuss this acinar to ductal hyperplasia that we see within 24 hours in the pancreas. So one of the other experiments that Sydney Moyer in my lab did is we obtained these mice, MST1 Cre. Okay, so MST1 is an acinar-specific transcription factor, and upstream of the MST1 promoter, there's a Cre transgene, which means that you can express Cre only in the acinar cells of the pancreas. Okay, and so Sydney worked out the tamoxifen conditions that gave you a similar percent recombination as our previous experiments with Mdm2 deletion in the entire pancreas. Okay, so similar percentage of recombination and similar activation of p53 targets. So here we use two of our targets, EDA2R and GTSE1, to measure p53 activation. And you can see in both mice where Mdm2 deletion happens in the whole pancreas, or mice where it only happens in the acinar cells, you have similar activation of these targets. So we felt we could do we could actually compare deletion of Mdm2 in the whole pancreas to deletion of Mdm2 just in the acinar cells. And we have absolutely no phenotype. So these pancreases look completely normal. Here we're measuring, we're looking at H&E sections, and here on the right we're measuring the immune component. And these pancreae are perfectly normal. So the take-home message here is that this acinar to ductal hyperplasia that we see is a p53 specific hyperplasia, but it's arising from signals outside of the acinar cells. So to me, this is a fascinating experiment because no one's ever noted that that the environment can affect the p53 response. And so we'll be delving into understanding this phenotype a little bit better. Pancreas is one of the tumors with 70-75% mutations in p53, and it always has this very compromised stromal component. And so maybe by understanding what p53 is doing in this physiologically important organism, we might be able to impact our understanding of p53 mutations in pancreatic cancer. Okay, for the okay, so I'm just going to check my clock to see how much time I'm doing. Well, okay, so I've shown you a lot of data where we deleted Mdm2 and I didn't show you data for Mdm4, but you see these p53 dependent physiological phenotypes, and that's all fine and good. It showed us how important this relationship is between these proteins, but what happens in human cancers is you've got high expressions of Mdm2. And this is just Yasmin Valentin Vega in the lab a number of years ago looked at Mdm2 levels in head and neck squamous carcinomas. And these are some of her beautiful pictures. So here's Mdm2 expressed very highly in a small region of this squamous cell carcinoma. Here it's expressed almost across the entire tissue. And then here's an interesting example of Mdm2 being expressed in the cytoplasm, not the nucleus. So we really don't understand what it's doing in the cytoplasm. But in all three of these experiments, p53 is wild type. Okay, so I think what this experiment and many others that people have done, again show that what Mdm2 is doing in these tissues is inhibiting p53 activity. Now, I also don't want to leave you with the notion that that p53 is the only Mdm2 target. Physiologically, it's the most relevant target because of the cell lethal phenotypes that we see. But in several experiments that my lab and Carol Privis's lab have done is we've tried to overexpress Mdm2 in normal cells to understand what it's actually doing with regards to transformation and tumor evolution. So here's what happens. So this is a normal control on the left. These are mouse cells, express a normal number of mouse chromosomes. And when we overexpress Mdm2, we see this incredibly abnormal chromosome instability. We can quantify the numbers of fusions here, and we have a huge number of fusions. We also have a lot of fragments. So in data from multiple labs, if you overexpress Mdm2 in a normal cell, the cell just, it dies. It can't continue to grow. So, and some experiments that are ongoing in the lab is okay, trying to understand, okay, so if we can't overexpress MDM2 in a normal cell, why do tumors have very high levels of MDM2? And one idea that we're working with is that there's some other mutation in those tumors that allows those tumors to survive with high levels of MDM2. And so if we could understand what else MDM2 is doing, we might be able to obtain a window of vulnerability to try to get the MDM2 overexpressing cells to implode. But the screens that we're doing currently are ongoing. Okay, then for the last few minutes of my lecture, I want to move over to our understanding of p53 mutations in breast cancer models. I didn't tell you earlier, but p53, well, I did tell you that p53 mutations are the most common, but really it's p53 missense mutations that is the most common type of genetic lesion. And so my lab and that of Tyler Jacks's lab have made germline mutations in p53 in animal models, and we show that these mice are tumor prone, but more importantly, in contrast to a p53 null mice, these mice have a high metastatic capability. So this here is our data from the 172 mutation, corresponds to the arginine 175 mutation, which is one of the hotspot mutations in human cancers. And here you can see a metastasis to the liver, and here stained with the p53 antibody, a metastasis to the brain. And this is in contrast to mice that have deletions of p53. So this really was the first example that suggested that expressing a mutant p53 was much more aggressive than not having p53. And in the field, we call this a gain of function. Mutant p53 is doing something in these cells to make them highly metastatic. So these are germline mice, and what we wanted to do is to generate somatic models, because these germline models represent Li-Fraumeni syndrome, which is an inheritance of p53 mutations, but that's a rare syndrome, and we really wanted to understand this metastatic phenotype in a system where the specific cell type has a p53 mutation, and the surrounding normal environment, the endothelial cells, the CAFs, the neutrophils, the T cells are all wild type for p53. So that mouse did not exist. Tyler Jacks made a beautiful mouse that has been used extensively in the literature that basically is heterozygous for p53. So the entire mouse is missing one p53 allele, and that allele can be converted to a mutant p53 in a tissue-specific fashion. So we didn't think that that was adequate enough to study the tumor stroma, tumor immune interactions because of heterozygosity of the p53 locus. So let me tell you a little bit about how we generated these mice and what our breast tumor phenotype is. Okay, so here's how we generated these alleles. So we call these a WM allele for wild type to mutant p53. So I'll show you in a minute. This is a wild type allele normally, and it's wild type because we inserted a cDNA sequence upstream of the point mutation. And this example is the arginine 172 mutation, which we generated previously. So what you have is a wild type p53. Here's your polynylation site. And then in a cream-mediated fashion, you can remove the wild type cDNA and basically reconstruct the locus, the R172 locus that we had studied forever in the germline of these mice. And because it took a very long time to make these animals, we actually decided to make a second hotspot mutation, and that's the arginine 245 to W mutation, which corresponds to the 248 hotspot mutation. So this is just shows you wild type initially, and then with cremated fashion, you make the mutant protein, and you can make it in a Cre-dependent manner. So this just shows you how normal those mice are. So here we're comparing wild type to heterozygous mice with the 172 or the 245. We stabilize the mutant protein in response to DNA damage. And when we look at the activation of p53 targets, P21 and Puma, they were activated to similar levels. We looked at the ability of these DNA damage to induce apoptosis, no difference between these two alleles in wild-type mice. And again, for the ability to arrest the cycle in mouse embryo fibroblasts, there's no difference. So these mutant alleles really represent, these conditional alleles, let's not call them mutant alleles, really represent a wild-type allele that can become a mutant. And this is the last experiment. I'll show you about the actual alleles. What we've done here is compared the 172 heterozygous mice, the 245 heterozygous mice, and wild-type mice to each other over more than 2 years to look at the tumor phenotypes. Mice is the age, just like people will sporadically get tumors, but what you can see is that there's absolutely no statistical difference between the 3 alleles. So for all practical purposes, this new allele that we generated expresses a wild-type p53 protein. Okay, so let me tell you about 2 experiments. One is our somatic, breast somatic model. And what we did is we used the R172H and the 245 alleles. We injected adenovirus Cre into the duct of the mammary gland. And then in addition, we use this TD tomato allele, which is also Cre dependent. So when we inject adeno-Cre, and I'll show you a picture in a minute, we basically make a mutant p53 and we label the cell red. So here's the control experiments to show that we do get recombination when we inject the Cre expressing adenovirus. Here on the left is a low titer injection, and you can see the red cells here. 1 to 5% of the ductal cells become infected. And then here on the right, you can see a high titer virus was injected into this gland, and about 50 to 70% of the ductal cells are injected. Okay. I also want to note that these mice are 50% Balb-C. Normally, the p53 field has used a C57 black 6 strain to study p53 tumor phenotypes, but that strain is resistant to breast cancers for some reason. And the Balb-C component brings in more sensitivity to breast cancers. And we really don't know the genetic reasons for this. Okay, so here's the data. Let me go through it in detail. So this is the 172 mutation expressed only in a few mammary glands, mammary cells. Low titer, we didn't see any tumors. High titer, we actually didn't see any tumors. This one tumor showed up post the 2-year end time point of the experiment. Because we weren't sure we were going to get any tumors by just making p53 mutant in a few cells, we also irradiated with a sublethal dose of radiation. This is not lethal to the mouse, but it does cause damage, and if the damage is in the right or wrong genes, it contributes to a tumor phenotype. So with a low titer, we begin to see tumors. We saw 1. At a higher titer, we now saw 4 tumors and 1 tumor metastasized. The 245 allele was a much stronger, had a much stronger tumor phenotype. With low titer, we saw 4 tumors. 1 of them was metastatic. With the high titer, we saw 9 tumors. So this is about 75%, and more than half were metastatic. So let me kind of summarize all the data that we've done with these animals. So first, let's just look at the tumor incidence, the R172H allele all by itself. And in these experiments, we only use 1 copy because we didn't want to do LOH, and we just wanted to figure out what would happen with the minimal number of alterations. So if you compare the 172 heterozygous to the 245 low and high titer, there's a huge number of tumors in the 248 mice. If we irradiated the 172, we got increased tumor incidence. And then this is the experiment where we did mutate the other allele, so exhibiting loss of heterozygosity, and we can see increased tumor phenotype with the high dose. And this is a comparison in the middle panel of the metastatic phenotype. And again, the 245W mutation was the most metastatic. And then we've looked at loss of heterozygosity. So the 245 mutation had variations in terms of LOH. About 50% of the mice showed LOH, and then others retained some or all of the p53 alleles. The irradiated R172H showed 100% LOH. So to us, while we don't understand why we see 100% LOH with this allele, what it says is that that wild type allele is very strong at inhibiting tumors in this 172 background. These are the breast tumor subtypes that we saw. The irradiated 172, we saw mostly triple negative breast. The R172 with the that lacks wild type p53, we saw mostly luminal B. And then here with the 245 mutation, we saw all three molecular subtypes. To me, this is a fascinating experiment because we've made one mutation, we've made a p53 mutation, and yet here in this sample with the 245, we see all three tumor molecular subtypes evolving. And so one of the experiments that we're doing now is trying to understand with this 245 mutation, what are the triggers to these different subtypes? You know, triple negative breast cancer is very hard to treat, but for example, these HER2 enriched tumors, you can treat with HER2 antibodies. So we're trying to understand basically the tumor evolution that initiates with this one p53 missense mutation. We've also wanted to, so the data I just showed you says that the 248 mutation is much more dramatic than the 175 mutation. So what I show here is a comparison of ovarian, lung, and breast tumors from people, and just looking at the kind of mutation that they have. And you can see in people that the 248 mutation has the worst outcomes. We couldn't do these data just for breast because the number of samples out there was not enough to give us significance. Okay, so one of the last experiments I'll show you here is just trying to understand tumor evolution, because we made a somatic model that developed different kinds of breast cancers that were highly metastatic. And so I'm really interested in understanding metastases in an in vivo physiologically relevant system. So what we did here is we took the 245 mutant animals, and we took two mammary tumors from these mice. We sequenced them in 3 different regions to try to understand a little bit about the heterogeneity. And then we sequenced 3 metastases from each of these tumors. Okay, and we sequenced them to an incredible depth. So what we have here then is the comparison of the primary to the metastases. And if we just look at mouse number 4, there is some overlap, and this overlap is considered an early gene signature, and then you see this these late sequences that come up, which is how where the metastases is now evolving when it gets into its metastatic site, which in this case was the lung. And so we can compare the early mutations of all 3 metastases, and you see early mutations in both experiments. So these are the mutations that were acquired in the primary and the metastatic lesion at the same time. But when you look at late mutations, here's all 3 mutations for mouse number 4, there's only 1 late mutation in common. So what these sequencing data indicate is that these metastases left the tumor very early during the metastatic process and then seeded and had additional changes. So this was the first suggestion that maybe metastasis, breast cancer metastasis driven by a mutant p53 is an early event. So to summarize this model, just briefly, we can make a p53 point mutation in just a few cells that become a tumor that intravasate, migrate, proliferate, and develop these metastases. We now have, I call it a little factory, but we just have these mice now developing tumors. We can isolate the circulating tumor cells. So what we're trying to do is understand the changes that occur for these cells to be able to survive in the blood, to home into an organ and then to develop. Okay, if I have a few minutes, which I have just a few minutes, I'm going to tell you about the other model that we made, because we weren't sure that making a p53 mutation in just a few cells was going to give us a tumor phenotype. So here we use K14-Cre, which expresses then a mutant p53 in all of the epithelial cells of the mammary gland. And this is a model that develops triple negative breast cancer 100% of the time. So again, this suggests that, you know, normal cell, tumor cell interactions are altering the kinds of tumors that come up. And in this scenario, we also had a Cas9 allele that is Cre dependent. So K14-Cre then allows us to make a mutant p53 in just in the epithelium of the mammary gland and to express Cas9. So we can use CRISPR technologies to begin to address vulnerabilities. Okay, so this the vulnerability that we examined in this model was whether these tumors were addicted to having a mutant p53. Okay, so here we use the adeno-associated virus that expresses a guide RNA that will delete p53. So the tumor burden before treatment with AAV in the control and the mutant p53 depleted tumors was similar. But you can see here in the purple line that those tumors that had depletion of mutant p53 survived much longer. This is just a picture of the tumor phenotypes, the control. You can see that it's obviously a tumor. These mice die very quickly post-identification of the tumors. And then you can see here with the depletion of mutant p53, this gland is looking more normal. Okay, this is a whole bunch of data for the individual mice. This is a tumor volume. The controls in green here. Once we identify the tumor, they just keep growing. The experimental cohort here in purple are animals that have recombined, have basically deleted that mutant p53 protein, and these mice live much longer. On the right is a tumor volume, so you can see that depletion of p53 affects the tumor volume greatly. And then I'll just point out these two samples in orange, which appeared not to respond to depletion of mutant p53. And when we look at these two samples in detail, they did not express a stable mutant p53. And so we think that these two tumors are actually, since the p53 isn't stable, the evolution that's occurring in these tumors is due to the absence of the p53 protein and not to a gain of function. Okay, so I'm going to stop there and just, I've thrown a lot of data at you, so I want to just summarize briefly. The first set of experiments really captured the exquisite sensitivity of the Mdm2 protein and p53 activity. We were able to identify this, the molecular response to p53 activation in vivo, identified numerous targets that are tissue specific. What are they all doing? I think we have our work cut out for us, because there's no way I can delete every one of these targets and see how important they are in vivo. And then last but not least, I showed you the generation of this novel mouse model that really allows us now to make a somatic point mutation p53 in any cell of origin that we want to. And in the breast model specifically, we had a highly metastatic phenotype that we're trying to understand in more detail. Okay, so my last slide is just the numerous people in the lab that have contributed to these studies. Let's see, Vinod Pant did the feedback loop studies. Roberto made the Mdm2 knockout. John made the Mdm4 knockout. Tamara did all the studies with the radiation and with Vinod to look at the p53 response. Sydney generated the conditional mouse to look at the acute activation of p53 targets. Ewan, terrific postdoc in the lab, who now has her own independent position, generated that conditional mutant p53 allele, and Donata is the one who's studying the addiction, and we're really wondering what the mechanisms is. Why are these tumors imploding when they no longer have a mutant p53? So with that, I'll end, and I'm glad to answer any questions.
Thank you so much. That was a wonderful talk. I'm going to ask people to put questions in the chat, but I wanted to ask you sort of from the therapeutics perspective.
Mm-hmm.
People have been very interested in compounds like Prima and Cody that assist with refolding of p53
Mm-hmm.
with disruptive mutation, and yet clinically those have been a little bit disappointing. Is much known about whether or not those refolded p53s are better, worse, the same as substrates for the Mdm2, Mdm4 degradation?
Yeah. So we've done a few studies using some of the drugs that are available, not many. Sort of my lab is focused on the genetics, because if we take out Mdm2 and Mdm4, we see a p53-dependent phenotype. But you know, it's very different. Genetic told us mechanisms, but the drugs are really, as you indicated, they're going to tell us whether they work or not. So I agree with you. I don't think the drugs are working very well, but you know, I don't know enough about those experiments to know, you know, how often the drug, with the level of activation. I think that that 7-gene signature that we identified would really help in those studies to try to understand what is the p53 response. I also think that the p53 response required will vary in different tissues. We just know from our Mdm2 studies that some tissues are just much more sensitive to increased p53 levels versus others. So I think that there's just a whole lot more work to do in the clinic to be able to understand the response.
Yeah. I think having a common set of, I mean, I think people have just looked at p21 over and over again, and that's probably very inadequate.
Right.
I see a couple questions, and we only have like a minute left. So first, Jeff Townsend wants to know whether or not you've considered looking at sequencing of much larger cohorts of tumors and multi-sample data sets to understand the temporal order of mutation appearances.
That's exactly what we're doing right now. I have a postdoc and a graduate student who have just generated a cohort of 100 mice, because we want to understand the sequence of events. We want to understand the different events that lead to the different molecular subtypes. So we are in the midst of those experiments, and we're going to do RNA-seq and DNA sequencing to understand both what happens at the RNA level. I think that it's critical. We need to understand what's happening at the DNA level, because I think that that's what gives rise to the different molecular subtypes. But I think it's the expression that's really going to tell us what's happening to those cells once they reach home to the liver or the lung. So we got all those are in progress.
Then Karen Anderson, who's my co-host for having invited you, wants to ask what your thoughts might be for a PROTAC directed against MDM2 to try to degrade that as a therapeutic strategy.
Yeah, I think there's two PROTACs we should be thinking about. One is MDM2, although somehow I think for an MDM2 inhibitor, I think you've got to target it to the tumor cell better, just because of the hematopoietic toxicities that have been seen with the MDM2 inhibitors. But I also think we should start thinking about potentially doing degrading mutant p53, taking it out of the picture to see what happens. When
Particularly with those gain of function mutations.
Yep, exactly.
We are over, there are more questions, but we are over the time. So I want to be respectful of and very appreciative of you having joined us today.
Super.
And
well, if anybody wants to ask me a question via email, but they should feel free to. I'd be glad to answer additional questions.
Great.
Mechanism. Okay, super.
Thank you.
Thank you very much, Barbara, for the invitation.